Good morning, dear students. This is your teacher Neela Melawat, and we will continue our chapter, that is chapter number 9, Biomolecules. Now, dear students, in the last video we have talked about the general structure of the amino acids, and we know that the amino acids basically they form these zwitterions, or as well when they are put into the solutions. Now, in this particular video, dear students, we'll be talking about the lipids. Have you heard the word lipids, everyone? Yes. So, lipids basically they are water insoluble, means they are not soluble in water. So, we can call them as non-polar solvents. Okay, they are non-polar, means they are not soluble in water, or we can say they are soluble in non-polar solvents like alcohol and benzene. So first property of lipids is that they are water insoluble. This means they are non-polar and they are soluble in non-polar solvents like your alcohol and the benzene. Now lipids basically, dear students, they contain three elements: carbon, hydrogen and the oxygen. But the amount of oxygen present in them is very, very less. So they are basically containing three elements, and only oxygen it is lesser in amount. Now lipids, if you observe around yourself or you observe yourself in the kitchen, you will find the ghee that we are using, the oils that we are using, that is all that will be included in your lipids part. Okay. Now lipids can be categorized into three types. One is your simple lipids, compound lipids and the derived lipids. So there is three categories of lipids. There are three types of lipids: simple, compound and derived. Now let's study each one of them separately. So first of all, let's take up the simple lipids. Simple lipid, dear students, they are made up of two main components. One is glycerol, another is fatty acids. So I can say there are two constituents of the simple lipids: glycerol and the fatty acids. Now, as I told you, simple lipids, they are made up of two components. One is your fatty acids and the glycerol. So glycerol is a alcohol-like component. Okay, clear? Okay. Now in this particular part, if you remember, we have talked about the structure of the amino acid, and we know that in amino acids, on one side they have, see, let's talk about the amino group. I'm just writing it here. So in the amino acids, if you remember, on one side they have a carboxylic group, on one side they have the amino group, third side they have a hydrogen atom, and on the fourth side they have an R group, which is called your variable group. Okay, which is called your variable group, right? Means this group, this R group, it changes from one amino acid to another. Now in case of your lipids, or rather I should say in case of your fatty acids, this R group this carboxylic group is attached to your R group. Okay, this carboxylic group is not directly attached with the carbon, but it is directly attached, it is attached with the R, that is your variable group. Okay, it is attached to the variable group, and thus we call it as RCOOH. We call this as RCOOH. Okay, so in case of fatty acids, if I'm talking about, I'm saying simple lipids, they are made up of two components: fatty acids and the glycerol. Now in case of fatty acids, remember that I'm comparing the amino acids with the structure of amino acids with fatty acids. We know in case of amino acid, carboxylic group is directly attached with the alpha carbon. But here the carboxyl group, it is attached with the R group. Okay, that is why we call it as RCOOH group. Like we can have an examples. First example we can take it as palmitic acid. Now if you look at the palmitic acid, just see. Can you see the palmitic acid? Yes. So palmitic acid basically contains 16 carbons. So in total, if you count these carbons, they're all 16 in number. Means 15 carbon are present, plus 1 carbon is of your carboxylic group. एक कार्बन जो आपका है वो आपका कार्बोक्सिलिक ग्रुप वाला कार्बन है, that is COOH. So 15 carbons plus your carboxylic group, one carbon atom from the carboxylic group, that makes a total of 16 carbons. So 16 carbons, it includes your carboxyl group carbon. Now second example we can take is arachidonic acid. It has 20 amino acids. So as you know, 19 carbon atoms are present, plus your one carboxyl atom we are counting here. So 20 carbon atoms, it includes your carboxyl carbon also. Okay. Now coming to the types of fatty acids. Fatty acids, they can be categorized into two types. One is saturated fatty acid, another is unsaturated fatty acids. Saturated fatty acids, the example is stearic acid. Just look at the structure of the stearic acid. Can you see? Just observe the carbon atoms. Just observe all the carbon atoms. We see that there is a single bond which is present between all carbon atoms. Right? So the fatty acids that contains single bond between all the carbon atoms is called as a saturated fatty acid, while the fatty acid, just look at the second example. Just look at the second example, everyone. Now just look at all the carbon atoms. So can you notice the difference here? Can you observe the difference? Very good, very good, exactly. See here, just look at here. There is a double bond present between the carbon atom. Right. So unsaturated fatty acid means, yes, the fatty acids where double bond is present, where one or more double bonds are present between the carbon atoms. Those are referred to as your unsaturated fatty acids. So we have two types of fatty acids: saturated fatty acids, unsaturated fatty acids. In saturated fatty acids, there are no double bonds, there are no triple bonds. Carbon is joined with a single bond. So the example is your stearic acid. Unsaturated fatty acid means where you will find double bonds are present. They can be one double bond, they can be more than one double bond. Okay, those will be referred to as unsaturated fatty acids. Example is your oleic acid. Oleic acid is an example of unsaturated fatty acids. Now coming to the second part, that is glycerol. So let's study glycerol. Glycerol is also called as trihydroxypropane. Okay, it's another name, or you can say its IUPAC name is trihydroxypropane. Now just observe. I have kept a picture for you. Just see. Can you see what is the color of the glycerol? Yes. Correct. It is basically transparent. So you must have observed this kind of component in your lab. So this glycerol is transparent. Now let's study its structure. Just see this. Just focus on the structure. It has 3 carbons. Okay, that is why we call it as propane, and all have a single bond between them. So 3 carbon atoms are present and all have single bond. That is why we call it as the propane. And there is, see, this group is called hydroxyl group. OH group is called as hydroxyl group. So there are 3 hydroxyl group, isn't it? On one, first group is present on first carbon, second on second, and third. So we have 3 hydroxy groups are present. That is why we give this name tri. Tri means 3. 3 trihydroxypropane. Okay. So one hydrogen atom has been replaced with hydroxy group. So that is why it is called as trihydroxypropane. Okay, everyone? So you will find that fatty acids, they're always esterified with the glycerol. Means fatty acids, they join with glycerol to form your simple fats or oils. Right? If you remember, I told you simple, you can say simple lipids, they are made up of two components, fatty acid and the glycerol. So now your fatty acid, okay, fatty acid, it will join with glycerol. Okay, it will join with glycerol, and this bond which is found between these two components is referred to as a ester bond. Okay, so fatty acid जो है वो आपका glycerol molecule से जुड़ेगा, add up हो जाएगा, और दोनों के बीच में जो bonding बनेगी, that is called as your ester bond. And because of the formation of this ester bond, simple lipids will be formed. Okay, simple lipids will be formed. Now just look at the diagram I'm showing you. This is a fatty acid which is, for example, it is palmitic acid. It will join with the glycerol molecule. Okay, so here, from here, what will happen? This OH molecule from the fatty acid, OH, and from here the H will be removed. So in nutshell we can say water will be removed. Okay, water will be removed, and after removal of the water, this O part, it will join with the carbon part, and thus it will result in the formation of the bond which is called as ester bond. Okay, and because of the formation of the ester bond, it would result in the formation of monoglycerides, diglyceride and triglycerides. Okay, now let's understand what does these bigger words means: monoglyceride, diglyceride, triglyceride. Let's see them. See, whenever one glycerol molecule joins with one fatty acid, we call it as monoglyceride. Okay, when one glycerol molecule it joins with one fatty acid, then we call it as monoglyceride. When one glycerol joins with two fatty acids, then it is called as diglyceride. And when one glycerol combines with three fatty acids, we call it as triglyceride. So I hope the structure, or you can say the name triglyceride is clear, or the glycerides is clear. So glyceride आपका तभी बनेगा जब ग्लिसरॉल प्लस फैटी एसिड आपका जुड़ जाएंगे, और दोनों के बीच में जुड़ने से जो बॉन्डिंग बनेगी उसको हम कहेंगे एस्टर बॉन्ड. तो अगर आपके पास एक ग्लिसरॉल है, एक फैटी एसिड है, तो देन वी कॉल्ड एज मोनोग्लिसराइड. एक ग्लिसरॉल प्लस टू फैटी एसिड्स हैं, वी कॉल्ड एज डाइग्लिसराइड. वन ग्लिसरॉल प्लस थ्री फैटी एसिड्स वी कॉल्ड एज ट्राइग्लिसराइड. ओके, नाउ लेट्स लुक एट द स्ट्रक्चर ऑफ द मोनोग्लिसराइड. देखिए, आई होप आपको ग्लिसरॉल का स्ट्रक्चर याद है. यस, ओके. So see, if you remember the structure of the glycerol, we have CH2OH. I'm just writing the structure. Then we have CH2OH in the glycerol, and again we have CH2OH, right? Now just compare this with monoglyceride. आप इस structure को monoglyceride से compare करिए. ये monoglyceride है, ये आपका glycerol molecule है. So if you observe, you will find this H has been removed. ये H यहाँ से remove हो गया है. Yes or no? And it has been attached by a fatty acid. This is a fatty acid. So you have one fatty acid is attached with your glycerol, and it forms a monoglyceride. Now just look at second. इस केस में आप देखेंगे दो हाइड्रोजन, वन यहाँ से एक हाइड्रोजन रिमूव हुआ है, एंड यहाँ से भी आपका एक हाइड्रोजन मॉलिक्यूल रिमूव हुआ है. सो इंस्टेड ऑफ टू हाइड्रोजन मॉलिक्यूल्स, टू फैटी एसिड्स हैव अटैच्ड इटसेल्फ. That is why it is called diglyceride. And just look at the third one. Again you will observe hydrogen, all hydrogen atoms, they have been replaced with three fatty acids. Your R1, R2 and R3, they represent your three fatty acids. So a triglyceride has formed. So monoglyceride when there is one fatty acid, diglyceride when there is two fatty acids, triglyceride when there is three fatty acids. Okay everyone? So R1, R2 and R3, these are your amino, you can see your fatty acids which are attached with the glycerol molecule. Now let's understand the structure. See, I'm discussing a structure of triglyceride. देखिए मैं triglyceride को structure को देख लेते हैं. See, ये if you look at this is your glycerol molecule. Okay, this portion, sorry. Let's rub it in, I'll do it again. This portion is your glycerol molecule, isn't it? This is your glycerol molecule. I hope you can notice it. CH2, if you remember CH2, I'm writing it here also, CH2OH, CH2OH, CH2OH. ठीक है बेटा जी. So CH2OH, CH2OH, CH, this is a glycerol molecule, right? You remember it? So in this structure, ट्राइग्लिसराइड के स्ट्रक्चर में देखिए ये जो पोर्शन आपका सेंटर में मैं हाईलाइट कर रही हूँ, this is of your you can say this is of your glycerol. Now this one, let's use different color. So this one is your fatty acid 1, second fatty acid, third fatty acid, and in between there is a ester bond which is formed, which is joining these two. दोनों के बीच में जो है जो एस्टर बॉन्ड बन रहा है, ठीक है ना, OCOO जो आपका बॉन्ड बन रहा है वो आपका एस्टर बॉन्ड है और वो आपके फैटी एसिड्स एंड आपके ग्लिसरॉल मॉलिक्यूल को ज्वाइन करता है. ओके बेटा जी. यस. ओके. So you have 3 fatty acids which are joined with the glycerol molecule. Now coming to the second category of lipids, that is called as compound lipids. Okay, compound lipids. See, in case of simple lipids we had 2 components. One was fatty acid, another is glycerol. In compound lipids we have 3 components. One is fatty acid, second is your alcohol like glycerol, okay, and third is some additional group will be present. So one, your fatty acid will be present, second is your alcohol like glycerol will be present, and third some additional group will be present. Okay, some additional group will be present. Just let's discuss, for example, phospholipids. So in case of phospholipids we have 3 components. One will we have fatty acid, second we'll have glycerol, and third we will have a phosphate group. So in addition to simple lipids, some additional group will be present here, and that is why we call it as phospholipids. Okay, we call it as phospholipids. Now these phospholipids basically they are involved in formation of cell membrane. Example is lecithin. Now let's understand the structure of the compound lipid. Again we will observe that this particular part is your glycerol. Okay, this particular part is your central part is of glycerol molecule. This is your fatty acid. So 2 fatty acids, and in addition to this you can see there is a phosphate group. So आपका glycerol molecule present है, आपके 2 lipids fatty acids present है, और एक आपका phosphate group present है. So these 3 together make up your compound lipids. So I hope you can differentiate between simple lipids and compound lipids. Simple lipids have fatty acid and glycerol, while your compound lipids they have fatty acid, glycerol, and plus some additional group that may be your phosphate as well. Now coming to the derived lipids. Derived lipids basically they are formed by the hydrolysis of simple lipids or the compound lipids. So when you hydrolyze the simple lipids or the compound lipids, it will result in the formation of derived lipids. So when you add water to simple or compound lipids, then it will result in derived lipids. For example is your cholesterol, and this is a structure of cholesterol. Now coming to the next part. See, we know that living organisms they contain a large number of carbon compounds. Living organisms के अंदर जो है बहुत तरीके के carbon compounds मौजूद हैं. Some of them they are the heterocyclic in nature. Okay, some of them they are heterocyclic in nature. They contain certain rings, and the best example of these heterocyclic rings is the nitrogen bases. Okay, nitrogen bases, the bases which are present in your DNA. So they basically are you can say are carbon compounds which contain heterocyclic rings. Okay. Now nitrogen bases, as I was talking about, we can categorize into 2. One is your purines, another is your pyrimidines. अब आपके जो नाइट्रोजन बेसिस हैं इन्हें हम 2 कैटेगरीज में डिवाइड कर सकते हैं. वन इज प्यूरिन्स, अनदर इज pyrimidines. Now just look at the structure of purines and pyrimidines and compare them. कंपेयर करिए, देखिए क्या क्या डिफरेंस है इनमें. If you look at the purine and pyrimidine, dear students, you will find that in case of purine you have 2 carbon nitrogen rings. यहाँ पर आपकी 2 कार्बन और नाइट्रोजन से बनी हुई रिंग्स है, ठीक है. देयर इज कार्बन एंड नाइट्रोजन रिंग, मीन्स रिंग्स हैं जिनके अंदर कार्बन और नाइट्रोजन प्रेजेंट है, एंड दे हैव 2 रिंग्स, 2 रिंग है इनके पास. इन केस ऑफ pyrimidine देयर इज ओनली 1 रिंग, एक रिंग है आपके पास, and this ring is made up of carbon and nitrogen. Coming to first purines में देखिए, there are 4 nitrogen atoms. आपके पास 4 nitrogen atoms available हैं. But in case of pyrimidines we only have 2 nitrogen atoms. So in purines we have 2 rings. In case of pyrimidine we have 1 ring. In case of purine 4 nitrogen atoms are present. In case of pyrimidine only 2 nitrogen atoms are available. Okay. So nitrogen bases, they are of 2 types, purines and pyrimidines. Now let's focus on all the types. Coming to the pyrimidine. Okay, the pyrimidine. Pyrimidine have 3 types of bases. One is cytosine, that is shown with C. Uracil, shown with U. Thymine, which is shown with T. Now out of these 2, cytosine and thymine, they are present in the DNA, while uracil it is present in RNA in place of T. देखिए DNA में जो है आपका साइटोसिन और थाइमिन दोनों अवेलेबल होंगे, ठीक है. RNA में थाइमिन नहीं होगा, इंस्टेड ऑफ थाइमिन वी विल हैव यूरेसिल. सो एट अ टाइम ओनली 2 pyrimidines will be present in nucleic acids, that is your RNA or DNA. So in case of DNA, cytosine and thymine will be present. In case of RNA, cytosine and uracil will be present. While there are 2 purines, adenine, which is shown with A, guanine, which is shown with G. Okay, so purines, they are of 2 types, adenine and guanine. Now coming to the pairing. Purines, they will always pair with pyrimidines. Purines जो हैं वो आपकी pyrimidine के साथ हमेशा pair होंगी. So A will always pair with T, and there will be double hydrogen bond between the 2. Guanine will always pair with G, and there is a triple carbon bond between them. दोनों के बीच में जो है 3 हाइड्रोजन बॉन्डिंग प्रेजेंट होंगे, हाइड्रोजन between G and C. So A will always pair with T, G will always pair with C. Now this in case of DNA. So in case of DNA this will be there. Okay, but in case of RNA what will happen? We will not have T. हमारे पास यहाँ पर जो है T नहीं होगा, instead we will have a U. Uracil will be present. So always remember it. Purines, they always pair with pyrimidines. So there will always, you will find 2 purines and 2 pyrimidines. Now coming to the next part, which is called as a nucleoside. See, this is a very important word, nucleoside. So we know that nitrogen bases, there are 5 types of nitrogen bases. 2 are purines, 3 are pyrimidines. So whenever a nitrogen base combines with a sugar, it forms a nucleoside. आपका नाइट्रोजन बेस जो है जब भी शुगर के साथ जुड़ जाएगा, एडअप हो जाएगा, then this will form a structure which is called a nucleoside. So adenine will join with sugar, it will form adenosine. अब आप देखिए स्ट्रक्चर देखिए, this is your you can say this is your pentose sugar. Can you see, this is a sugar, pentose sugar. It joins with adenine and we call it as adenosine. Second, guanyl will join with sugar, it will form guanosine. Again you look at this is your pentose sugar and the uracil. So this will form your uridine. Okay. So sugar plus nitrogen base forms nucleoside. So adenine will join with sugar to form adenosine. Uracil will join with sugar to form uridine. Thymine will join with sugar to form thymidine. Cytosine will join with sugar to form cytidine. And guanine will join with sugar to form guanosine. Okay. So nucleoside means nitrogen base plus sugar. Coming to another component which is called as nucleotide. See students, these two are very, very important. Students, many of the students, they generally, you can say, get confused with nucleoside and nucleotide. Nucleoside, nitrogen plus sugar. Nucleotide, just focus on nucleotide. So nucleotide is nitrogen, 3 components it will be have, nitrogen base, sugar and the phosphate group. So जब 3 चीज़ें मौजूद हैं, nitrogen base, sugar और phosphate, then we called as nucleotide. और we can say अगर हम न्यूक्लियोसाइड में, देखिए मैं लिखती हूँ यहाँ पर, न्यूक्लियोसाइड में फॉस्फेट ग्रुप ऐड कर दें तो हमारी न्यूक्लियोटाइड बन जाएगा. ओके, सो न्यूक्लियोसाइड joins with phosphate group, it will form your nucleotide. So adenine, it will join with sugar with phosphate group, it will form adenylic acid. Okay, it will form adenylic acid. Guanine plus sugar plus phosphate, it will form guanylic acid. Cytosine plus sugar plus phosphate, it will form cytidilic acid. Thymine plus sugar plus phosphate, it will form thymidilic acid. Uracil plus sugar plus phosphate, it will form uridilic acid. So nucleotides. Let's look at the structure. This is your pentose sugar. ये आपका pentose sugar है. ये आपका nitrogen base है, that is your adenine, and this is your phosphate group. So nucleotide is made up of 3 parts: nitrogen base, sugar and the phosphate group. Now coming to the next topic that we have, bio-macromolecules. So far we were discussing about the micromolecules. Okay, in micromolecules we have discussed 2 parts. One is your amino acids and second is your lipids. Both of them, they have been included in your micromolecules because their molecular weight is less than 1000 Dalton. Now let's come to the you can say biomacromolecules. See, I had in the beginning told you that although lipids, they are micromolecules, but they will always be present in the retentate part. ये जो है आपके retentate part में present होंगे, means they will be present in the acid insoluble part. Although it is a micromolecule, but it will always be obtained in the retentate part, right? It will always be present in the retentate part. Why? Because although lipids have a molecular weight less than 800 Dalton, जो lipids हैं आपके, उनका जो molecular weight है वो आपका 800 Dalton से कम होता है, ठीक है. लेकिन lipids जो हैं ये they arrange themselves and they form vesicles. जब आप किसी टिश्यू को ग्राइंड करते हो तो उसकी मेम्ब्रेन टूट जाती है और वेसिकल्स फॉर्म हो जाती हैं, और ये वेसिकल्स जो हैं दे आर अनेबल टू फिल्टर इटसेल्फ थ्रू द cheese cloth or the cotton that you will apply for filtration. So that is why lipids, they are not macromolecules, they are micromolecules, but they are always present in the acid insoluble fraction. तो वो आपको acid insoluble fraction में मिलेंगे, although they are micromolecules, but still they will be present in the acid insoluble part, which is your retentate part. Okay everyone? Okay. Now as I was talking about, let's start with the macromolecules. What are macromolecules? Macro means large in size. So the molecules whose molecular weight is greater than 1000 Dalton. वो चीज़ें जिनका वो molecules जिनका जो molecular weight है, okay, जो molecular weight है वो आपका 1000 Dalton से ज़्यादा है, उन्हें हम इस category में रखते हैं, that is they are macromolecules. Okay, so their molecular weight is more than 1000 Dalton and sometimes even more than 10,000. कभी कभी तो 10,000 Dalton से भी इनका molecular weight जो है वो बहुत ज़्यादा होता है. And there are three category of molecules. One is your proteins, second is polysaccharides, and third is your nucleic acid. So proteins, polysaccharides and nucleic acids, these are your important macromolecules. So we'll start with the proteins first. See, in general, if we talk about in a cell, there is 70 to 90% of water is present, and the proteins, they are only 10 to 15%. अगर एक सेल की composition को हम analyze करते हैं बेटे, so उसके अंदर it contains most of water, that is approximately 70 to 90%, and proteins, they are only 10 to 15%. The macromolecules that I was talking about, proteins, they're only 10 to 15%. Lipids, carbohydrates, you can see they are only 3% and 5%. So very less in amount, 3%, 2%. Nucleic acids, they are 5 to 7%. Ions, they are 1%. So the majority component is, in case of macromolecules, protein and you can say the nucleic acids, both of them, they are there in the macromolecules. So dear students, so far I think we'll be doing, you must read, you just go through these macromolecular part, the parts that I have done today. So you will make notes of this particular part, and you will read the page numbers 144 and 147. Since we have started with the concept of macromolecules, we'll continue it further in the next video. So in the next video, we'll continue with macromolecules, the topic of macromolecules, and we'll start with the proteins. And for today, you will be reading the page numbers 144 to 147. Make notes of this topic, and there are certain words which are highlighted in NCERT with black color. So don't forget to understand them, don't forget to write their definitions, and if there is any query, any doubt you feel, you can ask me, you can send your queries to personally to me on my number. Okay everyone, so that's all for today. Bye everyone.